My name is Laila, and this summer I had the opportunity to work on a research project in the lab of Dr. Daphne Gladman at the Toronto Western Hospital. Our lab focuses on better understanding mechanisms underlying the psoriatic diseases, specifically psoriasis and psoriatic arthritis. In up to 30% of psoriasis patients, they will go on to develop a more severe chronic inflammatory joint disease called psoriatic arthritis, or PSA. While we don't really understand what causes these diseases, we do know that there's a very strong heritable or genetic component. In our lab, we're especially interested in a phenomenon called the parent of origin effect. Now, what that refers to is that when psoriatic disease is paternally transmitted, that is transmitted from the father to the child, there's an increased risk of developing disease in the following generation. This type of an effect has been well characterized in other organisms, such as mice, and what often happens is that there's a change in the methylation of genes in the parent's germline, so their sperm or egg. Now, methylation refers to the placement of special marks on specific sites of the DNA that act as signs instructing the cell to make or express a gene. A change in these marks may cause a cell to make too much or too little of the gene, which can lead to disease. This change acquired in the germline of the parent is then transmitted to the offspring and contributes to the increased risk of development of disease in that generation. We refer to these changes as heritable epigenetic variants. Using this idea, our lab hypothesized that these heritable epigenetic variants may contribute to the parent of origin effect in psoriatic diseases. In a previous study, they identified several genes that were differentially methylated in the sperm of patients with psoriasis, psoriatic arthritis, and healthy controls. To show that these changes were inherited from the previous generation, they would need to show that the methylation changes are found consistently across different tissues in the same patients, as opposed to a spontaneous change that acquired in just the sperm cells of those patients in their lifetime. So the next step was to determine whether there were differences in methylation of these genes in a different tissue, and in our case, we chose to study their blood. In order to further validate our results, we were interested in determining whether there were any differences in the expression of these genes, possibly due to a change in methylation. So this was the aim of my project for the summer. By determining the expression of these genes in the blood of our patients and correlating this back to the methylation in the blood and sperm, we could provide more evidence that there are heritable epigenetic variants that contribute to this parent of origin effect. Overall, we identified genes which were differentially expressed in the blood between patients with psoriasis, PSA, and healthy controls. Additionally, we identified genes that had significant correlations between the methylation in the blood and the sperm. Interestingly, some of these genes are known to play key roles in immune pathways that contribute to psoriatic diseases. Overall, these findings help us better understand the epigenetic variations present in psoriatic diseases. As these genes may contribute to the development of psoriatic disease, in the future they may better inform the development of biomarkers and targeted drug therapies.